Great. Thank you so much, Jackie. And we welcome Carlos and Michael back on for the Q&A. We've had a bunch of questions that have come through, so I'll do my best to field them and get them to you guys. The first one is, I guess, just a lot of interest and participation in the trial. So how can parents find out if their child could qualify for trials like these? And a question about, you'd mentioned this was initially for Kabuki type 1, KMT2D, but if successful, when would you envision this being rolled out to KS2? Whoever wants to go.
Yeah, so I will take the first part of the how, and the answer to that is to please stay tuned. Okay, so we are going to we have been working very closely with KSF, our team, our academic collaborators, and ORYZON, to make sure that they know everything that's happening step by step, and with other amazing patient communities like All Things Kabuki and other people. And so we're going to make the information available to you as soon as we know the answers and can do that. And so, you know, please keep in communication with KSF, check their website, feel free to email them, feel free to email me anytime, and we can keep you up to date on that. But it's going to start, as Leila said, with older people and with a little bit older kids and adults, and then with KMT2D mutations. And that has mainly to do with just sort of the process of doing trials, right? You kind of have to show it in a certain population and then expand it to others. And then as far as the expansion, we don't know, but I'll let Michael and Carlos see if they want to add anything to that.
Sorry. Okay, so in terms of doing clinical trials, you know, it takes a while to set up. And so we're hopeful that in working with Jackie and the Johns Hopkins and the KKI team, that we'll start this trial sometime in the mid next year. Okay. And then that trial is about a half a year long, but it's not like everybody enrolls on day one. So it's going to take a little while for that trial to enroll and then read out. So I don't want to give specific dates, but I think it's safe to say that in probably about a year and a half after that time, we should know whether it's working in the first indication. And after that, then a decision will be made whether, based on that data, we can broaden it out into a wider population of either younger folks and or folks with a different mutation. Carlos, would you like to add anything to that?
No, well, a couple of comments. I think that you were covering most of it. Basically, we want to have, of course, a dialogue with the FDA, and to first of all with the FDA, and hopefully after with some European regulators as well. We hope that this trial might be also having some European patients. We believe that with the preclinical data package that we have, we might enroll patients younger than 18, and that would be really important. And that, I think, is a first step. So there are manners to expand the approach to the community. One is, of course, to go for the KTM6 mutation, but the other one is to see, and there was an interesting discussion yesterday, when is the best moment to start treating kids? No, what is the maximal clinical plasticity? And I think that this is also a route that we need to go, and we need to go step by step. So it is somehow is a bit slow. The whole process has to be slow because we have to basically ensure the safety of all the participants. And that comes also to the second comment I wanted to make, is that I think the most reassuring part of this proposition is that we have been treating patients really for very long time, patients that were in difficult situations, and the drug looks very, very safe. So this is something that it is not the ideal scenario when you find a drug which is already approved ready for repurposing, but I can say that we are in a sort of middle track here.
Yeah, that's great. And maybe a question actually for Dr. Harris is, you were involved with Takeda before, which was mentioned in the slide deck. And we had a couple of questions that came up from the audience in terms of a bit of the background there. So, what happened with the prospective Takeda asset, and really the community, I think, looking to feel confident about this project and ORYZON taking an LSD1 inhibitor through. So maybe a little bit of that context would be helpful for the audience.
Absolutely. So, so first of all, I just want to reiterate everything that Michael presented, and the ORYZON team has been so kind as to share with us some of the actual data that they have as far as the safety. And so I feel pretty confident that what we know thus far, that it is extremely safe. And so the answer to the question about Takeda is that, unfortunately, everybody does not have the same priorities, right? And so pharmaceutical companies have to answer to what makes the most sense for them at any given time. And and they also have privileged data, just like we as academics have privileged data, right? And so we do not know exactly what happened with with Takeda, and why exactly they dropped the project, because we are not privy to that data. That said, what is publicly available, there were no safety alarms from that perspective. So that's the good news. And what I could tell you just from what I know is that around the time that we were really, conversing with them about a trial, Takeda had a huge merger with another huge company called Shire. And the entire team that we had been working with was gone, and a new team was in place. And that happens in business and in industry, and they just weren't interested in the project, right? And sometimes that happens, priorities shift. And so, again, I don't know for sure, but that, timing wise, that's really how that all went down.
Well, sometimes there's a benefit to being smaller and more focused. So your small and brave team of 50, Carlos, we're all relying on you. There's a question for Dr. Ropacki here. So if the patient doesn't demonstrate problems with aggression in the first place, would the drug potentially have negative effects? So whatever the negative opposite of aggression would be, could it make the patient more hyper-passive or something else?
That's a great question. So what we have seen is that in populations that have been studied, either animals or humans, that don't have problems with agitation and aggression, it actually increases sociability. So it makes them more social, more wanting to interact with others. I don't want to say happier, because that's got kind of a connotation around it, but it makes you more enjoy and want to be involved with others. So that's what we've seen in the animals and the humans, such thus far, in groups that don't have agitation and aggression problems.
Great.
And adding to that, we didn't see any indication that we are producing with this mechanism of action sedation or I mean, any kind of we don't produce sedation.
Yes.
Exactly. I think that's at the root of the question. So thanks for addressing that.
Yes, thank you, Carlos. I missed that piece. So what we do know about the drug so far is that it seems to be, as we've said, safe and well tolerated. It works very well in agitation and aggression, increases sociability. There's been some evidence of pro-cognitive effects, so improvement of cognition. But from a safety perspective, unlike a lot of the drugs that are used in the psychiatry space, it does not cause unwanted weight gain. It does not cause sedation. and it does not cause what are called extrapyramidal side effects or things such as tardive dyskinesia. And I'm sorry, I'm using really big words there, but I don't know others to put that another way.
Sure. I think this whole audience for the last couple of days has now gotten a PhD in biology of some sort. So hopefully we'll keep up.
I wanted to say one more thing too, which is, keep in mind that based on the data that I was presenting and all of that, the targets for what we're trying to improve in Kabuki syndrome are specific to Kabuki syndrome. As you've heard multiple people, including me over the last few days, talk about outcome measures and things like that. We're looking to improve things that specifically affect Kabuki syndrome and think that perhaps we can based on the mechanism of Vafi. So just because you see changes in aggression or things like that in other disorders, that doesn't mean that that's what we're targeting in Kabuki syndrome.
Got it. That's really helpful. There are some questions around working memory versus long-term memory as it relates to the hippocampal functioning. Can one of you talk to that and kind of that distinction between the two?
I'm happy to do that, or if Jackie, if you'd like to take that on, please feel free as well.
Yeah, sure. So when we're talking about memory, we're talking about, and at least what we think from the data that we have for the mechanism of Kabuki syndrome, we're talking about getting those memories kind of stored into a longer function memory. There are other types of memory, like your working memory, where you're holding it there and manipulating it on the moment. And, one of the things that we're looking at when we talk about visuospatial memory is a little bit more sort of encoding that data into memory and being able to retrieve it later. And that's where the hippocampus and things come into play. And again, to reiterate, we are aware that a lot of people with Kabuki syndrome have very good memories. We are also aware it doesn't tend to be in the visuospatial space, which is, again, where you come to trying to design really good outcome measures, which Michael and I have been working very hard on to make sure that we're measuring something that we think is specifically improvable and important to improve in Kabuki syndrome.
And if I may add, just as an example, for those patients and family members in the audience, so working memory is more like, if you think of a computer, it has short-term storage, or you may want to say attention. So it's something that is in your immediate memory in a short-term storage, versus when you transfer it and it becomes a memory, that's more of your long-term memories. That would be my explanation of the distinction. One's more attentional, or a high-ordered attentional process, the short-term memory, and then the long-term memory is like when you transfer it and it becomes permanent in your memory.
And this next one you may not be able to answer, but there were some questions around, when taking the drug, how permanent or persistent is the change, and does it last beyond the administration of the treatment? So maybe you could talk about that in the preclinical side in terms of what you've observed, and then maybe what your hopes are, or what will be investigated as a part of the trial. Sure.
Carlos, do you want to talk about the preclinical, or would you like me to talk about that?
Oh, no, it's fine. I can do it if you want. I mean, no, we know from the preclinical experiments that we do have a long-lasting effect. And actually, we do have long-lasting effect on different outcomes. So we have been exploring also the, as Michael said, the anti-neuroinflammatory properties on specific multiple sclerosis models, which are very hard. I mean, these are not scales, or these are not basically memory measurements. These are basically measurements of physical impairment. And we see that we administer the animal the drug, and the protecting effect lasts for at least 1 month after the cessation of the treatment. And we have got similar results also in preclinical models of cognitive impairment, of memory recovery. It is more difficult to say the same in the clinical trials that we have done so far. But I mean, the basic mechanism of action, if we assume that we are changing the chromatin status and that this is basically tilting the transcriptional program towards a more open status of the chromatin, it should be long lasting. That's our idea. At least we will see when we get this more clinical data if that sustains. I don't know, Michael, if you want to add something
Sure.
on that.
So on the clinical side, there's the answer is, quite honestly, we don't know yet. And it's not that we don't know because of lack of wanting to know. It's an excellent question. It's a question that Carlos and I talk about all the time, because if you can induce a long-term effect, then you have a disease modifying drug. And that's very different than a drug that if you treat and you can affect a shorter term effect, it's more what's called a symptomatic treatment. But the reason I say we don't know for certain is that, fortunately or unfortunately, when you are doing trials with humans, you have to design the trial for the length of time that makes sense for the period of study for the drug and the specific disease under investigation. So you don't do trials with like animals, which you can follow those animals for a long period of time after you've given the drug and see how they do. In the humans, you have a very circumscribed amount of time. So for example, in Kabuki syndrome, we're talking about a half a year study, right? That doesn't mean that after a half a year that the drug will not continue to work. It just means that if you only study for a half a year, you only know how did it work during that half a year. And that's why I said this is a great question. And it's a question that we talk about all the time. But based on everything Carlos has stated, we do believe that if you induce this change, that it's not like an on or off switch, like an ibuprofen or a Tylenol, some kind of thing that you take and it works for a circumscribed period of time. The changes induced should be more long lasting than that.
That's really encouraging. Well, we have so many questions in 2 minutes of time, so I'm going to try something creative here and do a little bit of a quick fire on 2 questions. So if you could just answer as quickly as possible, 1 sentence or something like that. There was a question around how specific this drug is to the methylations that are connected to KMT2D. Presumably one wouldn't want to remove all methylations, correct?
Yeah, this is very specific. Actually, the molecular target of both enzymes are the same. So what the KMT2D is doing is exactly the opposite molecular job than LSD1. One is methylating and the other one is demethylating the monomethyl and the dimethyl forms of lysine 4 and histone 3. So it's very, very, very much the same.
That was a bit of a tough one to do very quickly, but you did great. And then the one I would like to end on is, could one of you please summarize in a couple of sentences what success looks like? So what benefits you'd hope to see in a Kabuki child following this drug's success?
This is for Jackie.
Yes. So the primary thing that we're looking at in this trial is cognitive neurodevelopmental manifestations. So looking at the specific areas that you've heard again and again tend to be pattern-wise difficult for Kabuki syndrome, visuospatial functions, potentially anxiety, and getting those better. And in addition, we're looking at some other things to get better, including things called biomarkers, which are like blood tests or other sort of more quantitative tests that correlate with that improvement, in addition to the functions of the immune system.
Great. Well, all named appropriately with Project Hope. So thank you so much for all of your time. I'll be introducing the next session, so I'll give you guys your time back. You can get off video and audio. Thank you so much.